Hello, intro to anatomy and physiology. It is Tuesday, April 14th, and we are here today to talk about the synapse in the neuron. This is basically where two neurons come together. Okay, so we are, this picture isn't in your packet, but I wanted to show you, this is an electron microscope image of what the synapse looks like. So we have, it's basically a connection between one axon's dendrites and then another axon. So it's basically this connection between two different types of neurons. So here we've got the axon of one neuron coming down, this little fibers here where they're connecting and basically coming in contact or touching with another neuron. And so the synapse is where the two come together. So they use the word presynaptic neuron for the one coming in because it's before the synapse. And then the postsynaptic neuron is the one it's touching because it's after the synapse. So it's kind of just this before and after term. So in your notes packet, we are on page 5 with this picture we're going to label and focus on today. So we're all talking about this connection between the two. So this guy right here coming in, this would be the presynaptic neuron, the one coming in. This one that it's touching here is the postsynaptic neuron, the one that is sending its signal away. So when we're zoomed in, this piece here is the presynaptic neuron, and then this one's the postsynaptic neuron. And this section where the two come together is called the synapse. All right, so just for the sake of the picture, I'm zooming in a little bit, but we're gonna label some things. So this box here, as I said, it's the synapse, this junction between two neurons. So where they come together, okay? And then we've got some structures here. This on the end, here's the cell body or the soma of my neuron. The little fibers coming off of it are called dendrites. Now, it's not called a dendrite when it's the tail end, like this piece right here of a neuron. Those are called the axon terminals, the end of the axon. And so right here, we actually have axon terminals coming in contact with the dendrite of the next neuron. And so this picture right here, this is showing an axon terminal. All right, so yeah, that label right here. So just referring to this whole end, kind of this, it almost looks like foot almost, if you would, sort of like stepping on or connecting into the next neuron, the postsynaptic neuron. So again, presynaptic neuron, postsynaptic neuron. All right, other things here to label. The space between the two is called the synaptic cleft. What I want you to note here is they don't actually touch. So a synapse kind of is in reference to a space a little bit. Now the space is so tiny. It looks like they're almost touching like on that picture I just showed you, but there is space in between these two neurons. They don't actually touch. So that space is called the synaptic cleft. And there's some important stuff that's happening, and we'll zoom in on the synaptic cleft here in a bit. But other things that are going on here, inside the end of this axon terminal, we have what are called synaptic vesicles. Think back to Bio 1-2 when you learned about the parts of a cell, and a neuron is a cell. It's just a uniquely shaped cell. Vesicles were something that stored stuff. So in this case, in an axon, the synaptic vesicles store neurotransmitters. And neurotransmitters are the chemicals that send signals to the body. And we have lots of different types of neurotransmitters. You learned a very important one in the muscular system. Acetylcholine, ACH, acetylcholine, was the main neurotransmitter that told your muscles to contract when it was time to contract. When neurons talk to other neurons, there's different neurotransmitters, not necessarily acetylcholine, that get sent. You've maybe heard of some like serotonin and dopamine and things like that are different types of neurotransmitters. But they store those chemical signals inside these little vesicles, these little pouches These little guys, hopefully you recognize, they are the powerhouse of the cell. Let's label those. I think you need to add a label on your page, but those are the mitochondria. And so these are all of the pieces here that I want to label. But I want to talk about what's happening here. And so there's different stages. And in fact, I'm going to use pink. Oh, sorry, one more label. I lied. This is pointing to the Schwann cell, just that covers that axon, which we'll talk more about in the next video. Those Schwann cells actually help speed up the rate of this signal that gets sent. And we call that signal that's being sent an action potential. So the presynaptic neuron, the first one, okay, is sending a signal down its axon. Remember, the signal always goes away from the cell body and towards the axon terminals. So I'm going to use pink here. Yep, we're going to use pink here to show these different steps. Like the black was what the stuff is. The pink is an action, what's happening on this picture. So the first thing that happens is an action potential. That's the fancy word we use for a little zap. It's basically the impulse, the electrical current, if you want to think of it that way, that gets sent. And our next video is going to really focus on what is an action potential. It's a little complicated, but we'll get through it. All right, so an action potential, a little impulse is getting sent down this axon towards the axon terminal. When the action potential reaches the end here, the axon terminal, the thing that signals everything is calcium. Much like calcium was super important for our muscular system, calcium ions, Ca2+, is important here. So calcium ions rush into the axon terminal, and then that in turn signals neurotransmitters to be released. That's step number 3. So if you can see, these little vesicles are fusing with the membrane, and those neurotransmitters that they're storing are getting released into that synaptic cleft. Once that happens and it joins up with these little channels on the bottom, then see the pink arrow here, a new action potential happens on my postsynaptic neuron. So we have an action potential on the presynaptic neuron and then an action potential on the postsynaptic neuron. Okay, so that's just steps 1, 2, 3, 4. To kind of show you it up here, okay, this signal down here, that would be step number 1. Okay, and then if we had another arrow going down here, that would be step 4. Okay, and then steps 2 and 3 are occurring right here at that little junction. So that's in short just the 4 main steps of what's happening here without getting into too much detail. But let's talk about what's happening when those neurotransmitters get released. So on the postsynaptic neuron, the receiving one, you have these little ion channels. Now think back to Bio 1-2. You have these little protein channels that can open and close, and they allow things in and out of the cell. In this case, these guys are, they only open with the right key. And in this case, the neurotransmitter here is the key that fits into a lock that opens the door. So in this case, there's no key, there's no neurotransmitter on the lock, and so the door is closed. But over here, you can see we've got the neurotransmitter, the key is in the lock, and now we've opened the door. So these are called chemical gated ion channels because they only open, the gates only open when the right chemical is there. And when they do open, a bunch of ions rush in. So that's chemical gated ion channel. So these little teal green dots here are the neurotransmitters. Here they're free floating in the synaptic cleft. Here it's attached, it's the key in the lock. And when that happens, okay, the neurotransmitter binds to the receptor on my gate. So here's a picture. That's the receptor without a neurotransmitter inside of it. Okay, that's basically like a lock without a key in it. But when the lock and key hook together, the neurotransmitters and the receptor, the gate opens and in rushes a whole bunch of not the neurotransmitter. The neurotransmitter does not go into the new cell. Instead, a whole bunch of ions, sodium and potassium, the main ones, are going to be rushing in. That rushing in is what tells the neuron to send its signal. And we're going to talk about that more in the next video, but it's all about ions coming into the new neuron. It is what that impulse, that when we think of like, ooh, our nervous system is like electricity running through our body. It's all these little, it's basically salt running in to our neurons that send a signal. So in order to send the signal, we need the lock to open the gate, and we get that lock when it gets released from the neighboring neuron. And this looks convoluted, but it is very fast. I mean, I just told myself to click my fingers and it did. And so we end up doing this incredibly quick. Our body's really awesome at how fast this actually happens. Now, after I've sent a signal and I don't want to send the signal anymore, that also needs to happen quickly. So what ends up happening is these neurotransmitters that are released in that synaptic cleft, they either just kind of float away or they get degraded. So there's special enzymes that break down the different neurotransmitters that are just hanging out in your body. So they eventually degrade, break down. They are removed from the lock, and then the door closes, and these ions no longer can pass in. That's the kind of the simple, basic understanding of what's going on at your synapse. Main thing again here, just these things in pink are the 4 steps. So if you wanted to, box them or highlight them, those are the steps of the synapse, like what's occurring, whereas everything in black is just the labels. That's all. You guys have a great day, and I will see you online next time. Bye-bye.